It's the day after Halloween, and you're worried about all that candy stuffed in your stomach. How are you ever going to digest it? The answer boils down to the proteins in your body. They help you with everything from digestion to dancing. But that begs the question, how are these proteins created? I'm Tia Vierling, and today I'm going to be talking about DNA transcription and translation, also known as the origin of proteins. DNA, or deoxyribonucleic acid, is probably something you've heard little about. It's basically your body in a nutshell. DNA contains the instruction manual for building everything from the roots of your hair to the soles of your feet. Any instruction manual has to instruct on how to build something, and for DNA, that something is proteins. DNA is made up of two strands of matching sequences of nucleobases. There are 4 types of bases for DNA: guanine, cytosine, thymine, and adenine. Adenine always matches with thymine, and guanine always matches with cytosine. To make proteins, DNA needs to collaborate with a similar molecule, ribonucleic acid, or RNA. RNA is a lot like DNA. The important distinctions for this video are RNA's single-strand structure and the fact that instead of thymine, it has the base uracil. First off, what do transcription and translation even mean in the context of DNA? Well, for transcription, think about video captions. A person speaking on TV, the DNA, has their words translated into captions by the RNA. Afterwards, somebody takes the captions and translates them into another language, just like how RNA's version of DNA's instructions are used to actually create proteins. For RNA's first job, transcription, DNA basically unzips its 2-strand structure to allow RNA to have sites to copy from. The RNA goes along one DNA strand and matches with its own bases. The DNA strand glues back together once the RNA is done, and that's it. Transcription is over. The new strand of RNA is called mRNA, or messenger RNA, because it is the messenger carrying the instructions needed for DNA translation. On to the real fun part. For translation, 2 types of RNA are needed besides mRNA. rRNA provides a ribosomal structure on which translation can occur, basically acting as a table where the instructions are carried out and the protein is built. tRNA is the protein translator. Each group of 3 bases on one mRNA strand makes up a complete codon. Each tRNA has a group of 3 bases to match the mRNA called the anticodon. Anticodons on tRNA are very special. They determine which amino acid is connected to which tRNA. For instance, a tRNA with the anticodon UAC would indicate that the amino acid connected to that tRNA is tyrosine. Different tRNAs with different anticodons show up to match with the codons on the mRNA strand. The amino acids of each of these tRNAs bond together as the tRNAs move from site to site along the strand. Once the sequence is done being matched, there's a stop codon to let the tRNA know that there's no more matches to be made. The amino acids already tied together make up a polypeptide chain, and that there is a protein. These proteins go on to help you in everything you do, even digesting that Halloween candy. And all your success is due to DNA transcription and translation. They're the processes that make you, you.